CRISPR stands for Cluster Regularly Interspaced Short Palindromic Repeats. CRISPR is originally used by bacteria to fight phage infections. Cas9 is currently the most widely known CRISPR experiments. The most commonly used Cas9 nucleus derived from Streptococcus pyogenes. As pyogenes, Cas9 only recognizes a pan sequence of NGG. The Cas9 endonucleus guided by a single guide RNA to direct site-specific DNA cleavage. The single guide RNA contains a 20-nucleotide guide sequence that determines the target site. Here's how CRISPR-Cas9 system works. Cas9 protein complexes with single guide RNA, recognize pan sequences on the non-target strand, and searches for the protospacer sequences that are complementary with single guide RNA. When CRISPR-Cas9 binds to the target DNA, it will result in a DNA double-strand break. Double-strand break can be resolved by either non-homology and joining called NHEJ or homology-directed repair called HDR. NHEJ works by randomly delete or insert nucleotides to repair damaged DNA. NHEJ can be exploited to disrupt coding and non-coding sequences through insertion and deletion. Although broken DNA can be repaired, the function of gene is disrupted. So, NHEJ DNA repair pathway is suitable for gene knockout. The other repair pathway, HDR. HDR requires a repair DNA template. We need to introduce a repair DNA template into cells to achieve HDR. The customized repair DNA template contains 5' and 3' homology arms that are totally complementary to the genomic DNA and the DNA sequences desired to knock in the genomic DNA. These sequences are flanked by homology arms. HDR repair pathway happens only after DNA double-strand break. After DNA damage, in the presence of a repair DNA template, homology arms complementary to the genomic DNA will facilitate the transfer of the DNA of interest into the genomic DNA. HDR allows the introduction of specific sequence changes. HDR repair pathway is a gene editing strategy to insert any interstate sequences. 2020, Emmanuel Charpentier and Jennifer Doudna have been awarded the Nobel Prize in Chemistry for their work on CRISPR-Cas9. The two scientists proposed several applications for the CRISPR genome editing, including changing the DNA of any species with extremely high precision, CRISPR clinical trials, and CRISPR-based COVID-19 diagnostics. Finding a treatment or cure for any disease is not that easy, especially for rare human diseases. About 80% of rare diseases have no cure. The most critical problem is the biology of how rare diseases occur remains to be studied. Rare diseases are mostly monogenic. Monogenic diseases describe a group of diseases caused by variations in a single gene. If we use next-generation sequencing to identify a variant of interest, or a novel variant in a patient, how that variant contributes to disease remains unknown. To mimic the diseases, we can edit the genomic DNA in either zebrafish, mice, or cells, using CRISPR-Cas9 precisely knock in patient-specific variants to generate disease models. Thus, models with knock-in patient-specific variants can mimic the diseases. Then, we can use these models to study the behavior phenotypes and verify the etiology and molecular mechanism behind the disease. We can get the answers to how the variant causes the disease. Identifying causative genes has important implications for accurate diagnosis of rare diseases and may even help for future treatments. CRISPR-Cas9 is a powerful tool to study the human diseases and improve disease research. CRISPR-Cas9 gene editing has been proposed as a therapeutic strategy for multiple human diseases, especially those with genetic causes. CRISPR gene editing makes the dream of curing rare inherited diseases come true. Monogenic diseases are the first targets for genome editing therapies, simply because editing one gene is easier than editing many genes. A single mutation produces non-functional protein and causes the disease. Gene therapy works by using targeted nucleases, such as CRISPR-Cas9, to correct copies of genes into the genome. This approach permanently removes a non-functional gene and replaces it with a functional gene in the patient's body. CRISPR-Cas9 gene therapies can be delivered through in vivo and ex vivo methods. In vivo CRISPR-Cas9 therapy describes the genetic material containing CRISPR-Cas9 and functional gene copies. that are delivered directly to the target cells in patient's body. It means the genes are precisely edited in the body. In 2017, Leber congenital amaurosis (LCA) was the first FDA-approved clinical trial of an in vivo CRISPR-Cas9 gene editing therapy. LCA is an autosomal recessive blinding retinal disease. About 50% of LCA patients are caused by mutations in RPE65. Under normal circumstances, when light enters the eye and hits the retina, stimulates the photoreceptors and converts the molecular 11-cis retinol to all-trans retinol. The formation of the all-trans retinol activates photoreceptors and initiates vision. After that, the RPE65 protein is required to convert all-trans retinol back to 11-cis retinol so that visual cycle can be restarted. RPE65 protein is produced in a thin layer of cells at the back of the retina, called the retinal pigment epithelium (RPE). If RPE65 gene mutations lead to a partial or total loss of RPE protein function, all-trans retinol cannot be converted back to 11-cis retinol, and excess all-trans retinol builds up in the retinal pigment epithelium. These abnormalities block the visual cycle, which finally leads to severe visual impairment beginning very early in life. LCA was incurable until gene therapy was approved. In this clinical trial, CRISPR-Cas9 components and functional copies of RPE gene carried by adeno-associated viral vector AAV2 was injected directly into the retinal pigment epithelium. The RPE65 gene is expected to be corrected and produce functional RPE65 protein in patients' retina, ultimately restoring the visual cycle. Ex vivo gene therapy describes the genome of specific cells from patients are edited outside the body and then reinfused back into the patients. A clinical trial began in 2019 using CRISPR-Cas9 corrected hematopoietic stem cells to treat red blood cell disease, beta thalassemia, and sickle cell disease. They are the most common monogenetic disease worldwide with autosomal recessive inheritance. Both are caused by mutations in the hemoglobin beta subunit gene (HBB). Hemoglobin, which carries oxygen in our bodies, is a tetramer made of 4 globin chains. The normal globin chain synthesis in erythrocytes. Before birth, the gamma globin chains combine with alpha globin chains to form a stable tetramer called fetal hemoglobin. Hemoglobin sustains oxygen delivered to tissue until birth. After birth, hemoglobin synthesis is blocked and replaced by beta globin. This converts to a combination of alpha and beta globin chains in adult hemoglobin. As you can imagine, patients with alpha globin chains suffer problems because adult hemoglobin requires functional beta globin. Switching process is mediated by BCL11A. BCL11A is a transcription factor that represses gamma globin expression and decreases fetal hemoglobin synthesis after birth. In this clinical trial, the strategy to treat the disease was not to directly correct the hemoglobin beta subunit gene, but to disrupt BCL11A expression and increase fetal hemoglobin. BCL11A is the target gene in this trial. In this clinical trial, hematopoietic stem and progenitor cells were obtained from patients. Through gene editing, BCL11A is normally expressed, but the regrowth-specific enhancer region of BCL11A is disrupted using CRISPR-Cas9 gene editing tool, resulting in blocked BCL11A gene expression. The gene-edited hematopoietic stem cells were reinfused back into the patients. It is expected that increases fetal hemoglobin formation. After birth, patients can rely on the fetal hemoglobin to replace the defective adult hemoglobin in red blood cells. This strategy can also be used to treat beta thalassemia and sickle cell disease. In addition to the applications of CRISPR system mentioned above, CRISPR can also be applied in diagnosis. Whether humans are infected by certain pathogens, this system is used to detect the presence of nucleic acid. As viral nucleic acid, bacterial nucleic acid. People are infected by viruses. Virus-containing samples are collected. In the presence of viral nucleic acids, these are extracted from samples. This RNA recognizes and binds to viral nucleic acid with high specificity. Specific targets could not be detected by CRISPR. In 2019, SARS-CoV-2 emerged and caused a global pandemic. Diagnostic assays have been developed for early diagnosis of a viral infection. Uses Cas13a to detect RNA sequence. Assay uses Cas12 to detect DNA sequence. CRISPR-Cas complex bonds to the target. Cas12 and Cas13 are active, binding nucleic acids non-specifically. This is called trans-cleavage activity. Linked by a short reporter sequence and a nearby quencher. It emits fluorescence when cleaved by an activated Cas enzyme. Fluorescence signal is detected by a fluorimeter. Button then labeled reporter in the reaction. The cleaved reporter can be captured on the lateral flow strip. This produces a visible signal. CRISPR-based diagnostic assay using DCAS9 is called assay. The Cas9 amino acid mutation in DCAS9 and HA40A to wild-type Cas9. Mutations in DCAS9 inactivate two catalytic domain of nucleus. Is incapable of cleaving DNA. Its ability to target DNA. After the assay, acids are reverse transcribed into CRISPR DCAS9 and bonds to specific biotinylated substrates. Labeled complex is detected by lateral flow assays. It's easy to read the result in a timely manner. Flow strip, anti-IgG rabbit antibodies are immobilized at the control line. Are immobilized at the test line. The sample is added to the sample pad, used in the test pad and the absorbent pad. The composites are captured in scaffolding with high affinity between bioteins. With anti-fan antibodies to both normal particles, and the strip shows two lines for a positive test result.